Hello and welcome to our webinar. The webinar is entitled, Does Nature Always Know Best? The Role of Regulatory T-Cell Epitopes in Biologics and Vaccines. My name is Annie de Groot. I'm CEO and CSO of Epivax and professor at University of Georgia Center for Vaccines and Immunology. Frances Terry will be joining me in this webinar today. She is Director of Analysis at Epivax. What is Epivax? Epivax is a 22-year-old company based in Providence, Rhode Island, with 35 employees. We provide access to cutting-edge tools, immunoinformatics tools, to a wide range of large and small biopharma companies and hundreds of smaller institutions and academic collaborators. Our corporate value is improving human health everywhere. We've had the privilege of working with great researchers all over the world for the last 2 decades, and today I'll be talking about work that we've done in collaboration with Brandon Koski at University of Kansas, Yoshi Takahashi at the National Institute for Infectious Diseases in Tokyo, Japan, and that work was continued at University of Georgia in collaboration with Ted Ross and Lenny Moisey. The outline of my talk today is to give you some background on T cells and immunogenicity, to talk about some tools that we use to differentiate T effector cells from Treg immune responses. I'll provide you with some examples, case studies in monoclonal antibodies as well as vaccines, and talk about Treg epitopes that we've discovered in biologic proteins. And I'll close with a short description of the role of Treg epitopes in antibody maturation, some recent work that we've been doing with Brandon Koski, and then Frances will provide you with an Esprit Demo. So here's a picture of the Esprit website. This is a website used by big pharma and mid-sized pharma all over the world. We're going to be focusing today on the antibody analysis silo, and we'll also talk to you about the homology analysis silo, which is where you find our Janus matrix tool, which we use to find tolerated or Treg epitopes in biologic proteins. To read more about the methods that we use to identify the immunogenicity of protein therapeutics, please look at this recent white paper by the Immunogenicity Focus Group at the American Association of Pharmaceutical Scientists. We also have a tool for vaccines, and so the computational vaccinology toolkit is called IVAX. It's been developed over the past 2 decades. Here's me at the outset of that research in 1998 on the cover of Inc. magazine. And 22 years later, we're happy to report that we've compiled a compendium of some of the more recent research and descriptions of the tools in a paper that was published in Frontiers in Immunology in April. So please take a look at that if you're interested in the computational vaccinology tools. The focus today is on both vaccines and biologics and how T cells determine what's foreign and what's self. Now, just to remind those of you who may have forgotten that T cell epitopes are linear peptides. They're presented on MHC or HLA molecules, and T cells are trained to recognize these linear epitopes in the thymus. So T cells are trained on self, and yet at the same time, they're able to recognize foreign pathogen epitopes or cancer epitopes that they've never seen before. Why is that? That is due to the ability of the T cell receptor to be a little bit wobbly when it's interacting with a linear peptide in the HLA binding molecule, and that enables a single T cell to recognize a self epitope and a foreign epitope. And that does have an impact on the resultant immunogenicity. So can we predict what the T cell sees? And does cross-conservation of T cell epitopes have relevance to immune response to vaccines and biologics? That's what we're going to talk about today. So some of the take-home messages are listed here. I'll be talking about how I hope that you'll be more aware of how common T cell epitope cross-conservation with self-epitopes, and here I'm talking about in the human, may be in nature. And I'll also describe the potential relevance of highly cross-conserved self-like epitopes to biologics and vaccine development. Hint, some of these highly cross-conserved epitopes may be Treg epitopes or what we call T-regitopes. And I'll also describe methods that are used to identify and validate Treg epitopes and how immune engineering them out of vaccines and into biologics may make those vaccines and biologics more effective. Now, whenever we talk about immunogenicity, we always talk about a lot of different things that can contribute to immunogenicity, but I will today be focusing on T cells rather than on the dose, route, and frequency of drug delivery or adjuvants or glycosylation or any of the above. Our focus today is on T cell epitopes and how they react to cell and foreign T cell epitopes. There's always a starting point, and so I think I do have to attribute all of the great work that was being done back in the 1990s by the Strominger group. So here is a reference to a paper by Roschke and Falk in Trends in Immunology, in which they described the HLA binding motifs that were used to identify and predict T cell epitopes way back then. And basically what they did is they eluted T cell epitopes off HLA molecules that were presenting antigens in culture. They then sequenced those T cell epitopes and found that there were recurring motifs that were present, suggesting that certain amino acids in the sequences were involved in binding that T cell epitope to the HLA binding groove. And so that became known as the HLA binding motif. And when enough of these HLA binding motifs were collected, people started predicting T cell epitopes directly from protein sequences. We were, of course, among the groups that were doing that. And the point that I wanted to make on this slide is that the quality of the T cell epitopes that are put into your model building has a huge impact on the accuracy of the tools. So the tool that we use is Epimatrix. The data that we use is highly curated before we start predicting T cell epitopes. We build matrices, so this is a matrix-based prediction algorithm that predicts linear sequences that bind to HLA. We can predict both class 1 and class 2, and I just want to mention again that HLA binding is not necessarily T cell response. What we're predicting is binding, and we have to go beyond that to actually assess immunogenicity. Now, all pathogens, all proteins are basically taken up by antigen-presenting cells, and the proteins are broken down into linear peptides and presented at the surface of the antigen-presenting cell to the T cell. And the amino acid residues that I mentioned before bind those peptides into the HLA binding pockets, and there is a face that faces the T cell receptor, which we'll talk about too. Now, of course, there are lots of different types of T cells, and yes, we can predict for all of them. Using as a starting point the sequence of the protein, we can identify CD4 or T helper epitopes that support the activation of the immune response, including this maintenance of the CD8 T cell response, as well as the development of effective antibody response by B cells. CD8 T cells recognize class 1 restricted epitopes. These are critically important for immune response to non-self. And yes, we can predict CD8 T cell epitopes as well. Prediction for Treg epitope is always a bit of a mystery because people always said that they were non-specific, meaning that they didn't really recognize a T cell epitope. But we asked why was there a T cell receptor? And we realized that these Tregs, particularly CD4 Tregs, were recognizing T cell epitopes on the surface of antigen-presenting cells, just as T effector cells were. So why is it that some T cell epitopes are not immunogenic, don't generate an effector T cell response? Why is it that some epitopes generate a tolerogenic immune response and activate regulatory T cells? The HLA binding affinity turns out to be the same. The ability of the peptide to bind in the HLA binding groove and the presence of an HLA binding motif is basically the same. So if you think about what I've mentioned before, one comes to the conclusion that the T cell receptor face must be relevant. And that was actually something that we discussed with our FDA colleagues, and I'll talk a little bit about Amy Rosenberg's theory about factor 5 and factor 8 and cross-reactivity between factor 5 and factor 8 at the T cell receptor face a little bit later on in this talk. So if there's more to T cell epitope than HLA binding, and the TCR face is key to understanding the immune response, then of course we have to build tools that enable us to look at that T cell receptor face. And we can do that by looking at how the peptides bind in the HLA binding groove. Clearly there's a face that faces down, and there are amino acid side chains that bind into pockets in the bottom of this molecule, which is shown here with 2 alpha helices framing that surround a floor where the binding pockets are found. And here's a cross section of the same interaction of the linear peptide binding to an HLA molecule with its amino acid surface pointing up to the T cell receptor. And what we can do is we can identify the amino acid side chains that point up to the T cell receptor and those that point down, and start comparing across T cell epitopes which T cells might be presenting the same face to a T cell. So a T cell trained on self might start recognizing or cross-recognizing a cross-conserved T cell epitope that presents the same face at the T cell receptor surface. So now that we've convinced you that we can predict binding, can we actually start looking at T cell epitopes and pathogens that are conserved with self? It turns out that there are many of these T cell epitopes, not a huge number, but significant number, especially in pathogens that like to be commensal. And these T cell epitopes that are found in pathogens are not just conserved with any self epitopes. It makes sense, if you think about it, that these T cell epitopes with which they might be conserved could potentially be regulatory T cell epitopes. And this raised the next question, which is, what if those Treg epitopes are actually in vaccines? Then the immune response might be modulated by the recognition of a Treg of that particular sequence, which would down-modulate immune response, reducing antibody response, reducing CD8 T cell response. And so it became pretty important to start looking at T cell epitope cross-reactivity and how it may influence immunogenicity. We were actually starting to think about the influence of Treg epitopes and how to discover them because we had been working on what we call Tregitopes. These were peptides discovered in immunoglobulin G that were promiscuous HLA binders. They bound to multiple different HLA class II alleles so that they could be presented widely in different human populations. And they were found right outside of the CDR region in immunoglobulin G, which suggested that they were there to actually dampen down immune response to foreign epitopes that were generated as the antibody matured. And so these Treg epitopes were isolated, we synthesized them, and in collaboration with David Scott, we published our first paper in 2008 showing that they induced natural regulatory T cells and probably were relevant to biologics development because people were making monoclonal antibodies. So the test, of course, was to examine whether they could be associated with outcome, what would be the outcome, anti-drug antibody responses, ADA responses. And here I'm just showing you a quick picture of a retrospective study that we did with a number of clinical phase antibodies, looking at their sequences, identifying their T effector epitopes, identifying their Treg epitopes, and trying to determine which model could be used to predict immunogenicity. As shown on the left here, if we just count up the number of T cell epitopes that are present in an antibody molecule and try to correlate that with the immune response. So the raw Epimatrix score is on the x-axis, the immunogenicity as reported in the clinic is on the y-axis. You can see that the correlation is pretty poor looking at the R-square value. If, however, you correct for the presence of Treg epitopes and discount their scores, you get a T-regitope adjusted Epimatrix score, which has a much better correlation with clinical observation of immunogenicity. And the equation that we use to predict now, prospectively, the immunogenicity of any antibody molecule is shown here, where we plug in for X, the T-regitope adjusted score, and we can output the Y, which is the predicted immunogenicity of that molecule. Now, does this actually work? Well, I just want to point out a few examples. Here you can see a very nice publication by our colleagues at Chugai, who have been using Epimatrix and T-regitope adjustment for years. Here you see in their paper in Figure 7 a comparison of the Epibase non-T-regitope adjusted analysis of this original antibody and then the final product that they put into the clinic, and the Epimatrix assessment of the same, showing that when T effector epitopes were removed, the Treg epitopes were preserved, and that molecule ended up not being an immunogenic molecule. Another example of a non-immunogenic molecule produced by the Chugai group using the Esprit toolkit is shown here. This is the antibody emicizumab, which is a bispecific used for prophylaxis of bleeding in hemophilia A. And it is a non-immunogenic molecule, highly successful in the clinic with very little signal. This is just looking at a snapshot of the CDR2 region where you don't see a lot of 9 MERS in the CDR region that have high scoring or dark blue binding regions for any of the HLA class 2 alleles shown at the top. So this suggests that this epitope, this molecule would not be very immunogenic. And just to give you a contrast, here's an Epimatrix analysis of the CDR2 region of the GSK anti-IL-6 antibody that was immunogenic in every single individual that was tested in their phase 1 study. It's interesting to note that they do not use Esprit and mention that the in silico analysis was clean. To my eye, the CDR2 region is packed full of potential immunogenic T-cell epitopes, and so I'm really surprised by that statement. Okay, so that's just showing you T effector epitope. How do we discover Treg epitopes like T regitopes or human-like epitopes in biologic proteins? Well, the Janus matrix concept basically is to look at both sides of the T-cell epitope. Let's find epitopes that bind to HLA molecules by looking at the amino acids that bind into the HLA binding groove illustrated in blue in this cartoon. And then let's isolate the upward-facing amino acids in that same sequence and compare those to other epitopes that may be in the host genome. In this case, we're going to be focusing on human. So how do that? We visualize the concept using the Cytoscape interpretation of Janus matrix output as shown here. This shows you the epitope networks of epitopes found in the human genome that might be associated with an epitope in a biologic protein. Here, for example, say we have found a promiscuous binding 9-mer from a biologic protein like a monoclonal antibody. And what we're going to do is isolate each HLA binding peptide and examine its TCR phase for epitopes that have the same sequence in the human genome. So the TCR sequence is identical. The HLA binding side of this peptide might be slightly different, but it has to bind to the same HLA. Here you can see in this example, we found 6 epitopes that are not necessarily identical in sequence, but have the same TCR phase and would be recognized by the same T-cell epitope, T-cell or T-cell receptor. In contrast, you can see in this same promiscuous T-cell epitope, there is one HLA binding motif that's not cross-conserved at all with any of the human genome T-cell epitopes, which suggests that it's potentially immunogenic due to low cross-conservation with self. It would be viewed as foreign for the person who has that specific HLA DR. And so this is the visual map. How do we apply this? We first described this discovery and this methodology in a paper called The Twofaced T-Cell Epitope, first author Lenny Moisey. This was work actually done by Andreas Gutierrez when he was a postdoctoral student or doctoral student at University of Rhode Island with me. Now, a future application is what I mentioned previously with Amy Rosenberg. We had this discussion about what is it about T-cell epitopes that makes them regulatory, and she had proposed, as we had, that perhaps the prevalence of the protein in circulation and the presence of a T-cell epitope that's constantly educating the immune response to be tolerogenic might be one factor. So she had suggested that we take a look at factor 5 because of its homology with factor 8. And she said that the reason she was interested in this was that perhaps the reason why some hemophiliacs did not react to factor 8 epitopes was because they were cross-conserved with factor 5, which continued to be circulating in hemophiliac blood, even though they lacked the factor 8 molecule. So we were intrigued by that hypothesis. We used Janus matrix to find some T-cell epitopes that were cross-conserved between factor 5 and factor 8, and then tested them in vitro assays where we actually measured immune response to tetanus toxin and how much the T-cell epitope from factor 5 could actually suppress that immune response. So that paper was recently published. If you're interested in the tetanus toxin bystander assay that we use to validate Treg epitopes, please take a look at this paper. So going back to human pathogens, we actually think that this is one other way of finding Treg epitopes, because some human pathogens appear to have introduced into their own sequences T-cell epitopes that allow them to camouflage themselves from immune response. And this is much more common, as it turns out, in pathogens that are commensal. So here's a paper where we looked at 152 human pathogens. These are viruses, and we examined each of the 15 MERS in their entire genomes. This was work done by Lou He and Chris Bailey Kellogg at Dartmouth. And what they showed is that those pathogens that tend to be commensal have many more human-like epitopes and many fewer T effector epitopes in their genomes, just using this mass analysis approach, than pathogens that are hit and run, so to speak. So Ebola, smallpox, pathogens that just kill humans and don't like to hang around and live inside of them, having camouflaged themselves to the immune response. So this was an interesting observation. To give you a couple of examples, in HIV, we found a promiscuous HLA-DR restricted T-cell epitope that's found in every subtype of HIV-1 circulating in the world in the envelope protein, and it is highly cross-conserved with the human genome, as shown here, and in particular, it's cross-conserved with the HLA class 1 molecule. So we thought that was very interesting. We also found in hepatitis C another promiscuous HLA-DR binding epitope, highly cross-conserved across subtypes of hepatitis C, and this one is conserved with actually apolipoprotein B, and was shown to be a Treg epitope in vitro, and its homologue, the apolipoprotein B epitope, is also tolerogenic. Okay, so how does this apply to biologic proteins? Well, one application is to look at host cell proteins. This is an interesting exercise, particularly now that we're working on vaccines and we're concerned about the potential for host cell proteins in the vaccine product to induce potential immune responses that are off target. And so here, for example, is an analysis of a biologic product in which there were several host cell proteins. Their parts per million are shown by the size of the dot. And we've analyzed them by putting them in a two-dimensional graphic where we have Janus matrix score on the x-axis and Epimatrix score on the y-axis, and we can localize these host cell proteins in different quadrants. A quadrant, for example, where you have lots of T-cell epitopes that are foreign in the host cell protein, say it's a CHO protein, for example, that might potentially trigger an immune response, whereas you have other host cell proteins that are highly homologous to human and that have low T-cell epitope content. You can imagine that those might either be tolerated or completely non-immunogenic due to the lack of T-cell epitopes. So we can do host cell protein analysis this way, and we can also analyze cancer proteins and cancer epitopes in the same way. I don't have time to discuss that today with you, but please look for our tool called Answer that we use for personalized cancer vaccine development. Now let's go to the H7N9 case study. This is an interesting story that started in 2013 when a new avian influenza emerged to which human beings had no protective antibody response. And there was a lot of concern about this. And so we did an analysis where we looked at the T-cell epitope content of the critical antigen, which is HA. And we also looked for Treg epitopes in that antigen and found them. So we actually published a paper which said, we think that this particular HA antigen, which would normally be produced as a vaccine for this influenza strain, might be very poorly immunogenic. And little did we realize the risk that we were taking, because actually at the time, nobody had made a vaccine to this pathogen, and what happened if we were wrong. It turns out that the antigen was non-immunogenic, that it was very poorly immunogenic in human studies when compared to H1N1. People who got a single dose of the H1N1 antigen without adjuvant, over 60% have antibodies after the first dose, whereas only 20% had antibodies after the first dose of the H7N9 HA protein. And so that was a prediction that worked. Now, how can we fix that problem? This is work that was done by Lenny Moisey and that he continues in collaboration with Ted Ross at University of Georgia. So what we proposed to do was to add effector T cell epitopes to make the vaccine antigen more immunogenic and take out that Treg epitope that we had discovered in the H7 protein. So we did that simply by swapping 3 amino acids as shown on this slide. We found that the H7 Treg epitope, which was highly cross-conserved with the human genome, was only 3 amino acids different from a promiscuous class 2 restricted T effector epitope that had been found in H3 and published previously. So we swapped out the amino acids and produced a new H7HA that we called OPT1 and tested that in a humanized mouse model with Yoshi Takahashi of National Institutes of Infectious Diseases. What we saw in paired mice, each mouse having received peripheral blood cells from a human being, and these are JAK2 knockout mice that can receive PBMCs. What we saw actually in these 8 donors and 16 mice was that the wild type H7 antigen, when used as an antigen, did not induce a very strong antibody response in the mice when used as a vaccine, whereas simply changing 3 amino acids increased the overall antibody titer by 5-fold and the B cell response by 20-fold in this humanized mouse model. That was published in Scientific Reports in 2017. And then we've gone on to do challenge studies looking at pre-immunizing mice with H3 because you want to have memory to that H3 epitope and also measure the immune response once you remove the Treg epitope. Here you can see on the left-hand side the antibody response to the optimized antigen of H7 in these mice. These are HLA-DR transgenic mice that have been pre-immunized with H3, and the wild type response to that H7 antigen. Sorry, the HLA-DR mouse responds to the wild type H7 antigen. And these are the mice that had just the pre-immune H3 exposure, but no boosting with an H7 antigen. When these mice were challenged with the very virulent H7 strain, you can see that the optimized antigen, where we had removed a Treg epitope, inserted a T effector epitope, was much more effective at protecting these mice from live challenge than the wild type antigen, which did not have the Treg epitope removed. So that's a very important finding. We're going on with Lenny and others to develop even better H7 antigens. And it just proves the point that one can engineer vaccines to increase antibody production and remove Treg epitopes that attenuate antibody response. Now, just in your mind, flip this and think about what happens if you introduce Treg epitopes in biologic proteins. Clearly, you can reduce anti-biologic antibody response. Okay, so here are some papers that describe the work that I've just shown you. First, the paper by Ri Lu discovering the H7 and 9 Treg epitope in H7 and 9, and then the immune-engineered H7 and 9 flu study that was performed by the group at University of Georgia in collaboration with Epivax and Lenny Moisey from Epivax. I do want to mention other applications of Janus matrix, so I'll quickly mention this CAR T study. In this particular study, there was a problem with a CAR TCR targeting the HLA-A1 peptide of MAGE-A3. You may have heard of this when these authors got to the phase 1 trial and infused CAR TCR T cells. They had an unexpected off-target response to cardiac tissue. And when they did an alanine scanning approach, they recognized that the MAGE-A3 had similarity to the titin protein of cardiac muscle that may have contributed to the deaths of 2 subjects in the phase 1 study. We applied the Janus matrix tool to the MAGE-A3 epitope. It's a class 1 epitope, and the results were quite striking. As you can see here, we looked at the MAGE-A3, found the class 1 epitope, and examined the cross-conserved T cell epitope in the human genome. And you can see that there were few, most of them were melanoma-associated antigen MAGE-A3, but there was one cross-conservation with human titin protein. And so I think that is another application of the Janus matrix tool that is worth bearing in mind. Okay, well, so having alarmed you, now I'm going to try to get you not to worry too much. Actually, cross-conservation with self is fairly rare. We recently did this analysis at the request of some colleagues at the FDA who were concerned about potential cross-conservation between T-cell epitopes found in pathogens and human tissue. So first we started with a set of 100,000 random sequence proteins generated using the XPASI protein sequence generator. We found 10,000 random T-cell epitope ligands in those proteins, and then we analyzed them for cross-conservation with the human genome. Only 5% of those 10,000 peptides had a Janus matrix score of 3 or greater, which we consider human-like and potentially tolerated, and only 1% had a Janus matrix score of 5 or greater, which suggested that they could actually be tolerogenic. That's our threshold for potential tolerogenicity. So cross-conservation with self is not that common in a set of random sequences. How about pathogens? Here, what I did is I downloaded all of the T-cell epitopes in IEDB. I'd say this was probably a few months ago, and I restricted that search to looking at HLA class 2 restricted epitopes that had been shown to be positive by some measure in the IEDB database. So these are what we would call validated human pathogen T-cell epitopes from IEDB. And of those epitopes, 10% had a Janus matrix score of 3 or greater, so potentially tolerated, and 4% had a Janus matrix score of 5 or greater, suggesting they could be actively tolerogenic. So there is slightly more cross-conservation between pathogens and self, and so we haven't dissected out which pathogen is which in this analysis, but it is just kind of a big data crunching exercise. It's worth evaluating that cross-conservation when designing vaccines and, of course, biologics. Okay, so now we're going to go on and we're going to talk a little bit about Treg epitopes and biologic proteins. This is also work that we've been doing with FDA. Here, what I'm talking about are biologic peptides. There was a recent draft guidance that mentioned that we should be looking at T-cell epitopes in impurities that result from peptide synthesis. By impurity, we mean peptides that are not the drug product, the API, the drug itself, but they are actually peptides that are present in the drug product that have either an amino acid deletion, insertion, or modification that results in the sequence not being the same as the actual drug. And so we've been working with the Office of Generic Drugs to look at drug impurities, and we found actually that it was quite interesting that some of these self-peptide drugs like GLP-1, liraglutide, and glucagon, and also teriparatide, which is shown here, have sequences that look potentially highly immunogenic. Here you see a 9-mer frame with predicted binding to multiple HLA class 2 alleles that could be highly immunogenic, as shown by the overall score that is quite high. But when we examined this particular peptide in parathyroid hormone, or teriparatide, the drug, we found that it was highly cross-conserved with the human genome with a specific protein called tubulin, which is present in every cell in the body. So we hypothesized that it could be tolerogenic, and we were pleased to actually see in our tetanus toxin bystander suppression assay, where we put in a peptide like a teriparatide peptide with tetanus toxin and examine how peripheral blood cells respond to the tetanus toxin with a memory response, and then how much that putative Treg epitope or T-regitope could suppress that immune response. And as you can see in the results from the teriparatide study, that we actually found that the teriparatide peptide drug performed much like a T-regitope. Here we're using T-regitope 289 from immunoglobulin G and factor V 621, and you can see that compared to tetanus, each of these putative Treg epitopes suppresses the memory T-cell response in a dose-dependent manner, both in terms of proliferation and activation when we put them in culture together. And so that was an exciting finding, suggesting that some peptide drugs might have self-like properties that we can uncover using Janus matrix. We then looked at some potential impurities. And so here you can see the human version of teriparatide with a high score of 16, a Janus matrix score of 4, the number of donors responding in a set of 30 was about 19%. And when we mutate that peptide to remove the human face of the T-cell epitope, reducing its Janus matrix score but keeping its high Epimatrix score, we see an increase in the overall immune response as illustrated in this slide here. As Janus matrix human homology score goes down, immunogenicity goes up. So we think that's very exciting, and it also tells you, same thing is probably true for cancer epitopes. When you're designing a cancer vaccine, there might be epitopes that are highly homologous with the human genome, introduced perhaps by the tumor to escape from immune response, and this same type of analysis with Epimatrix and Janus matrix can enable us to make better cancer vaccines. All right, so last but not least, I wanted to mention antibody maturation and Treg epitopes, and then I'm going to turn this over to Francis. So this is work we did with Brandon Takehome, who has developed methods for sequencing individual B-cell, heavy chain, and light chain CDRs. And when he told us about this at a meeting at the FDA, we're very excited because we really wanted to look at what happens to T-regitopes as antibodies mature. So here we're going to use somatic hypermutation as a proxy for antibody maturation. And as you know, all of these cells are living in the B-cell follicle. The B cell is trying to mature and improve its affinity by introducing mutations that allow it to bind better to the target antigen. And there are both T helper cells and Treg cells that are involved. So what is the role of T-regitopes in that gamish? Can we look at the sequence of antibodies as they mature for a single individual, which is the database that Brandon made available to us, and determine what happens with Treg epitopes as antibodies go through the maturation process? To do this analysis, which was really done by Matthias Gutierrez and Andres Gutierrez, we looked at T-cell epitope content using a specialized score that we call ITEM for individualized T-cell epitope measure. We only use the HLA of the subject that is producing those antibodies. We can then hone in on T effector epitope content by adjusting for Treg, take the T-regitopes out, just look at the T effector epitopes, and we can further refine the analysis by removing any epitope that is highly homologous with self by doing a Janus matrix adjustment. And then we can just look at T-regitope content, so since we know what the T-regitopes are in antibody molecules. Do they increase with somatic hypermutation, or do they actually decrease? And we initially hypothesized that T-regitopes might decrease. And that was kind of interesting because the first data that was reported to us did show that the overall T-cell epitope score declined as antibodies matured. So here's somatic hypermutation on the x-axis and the overall Epimatrix score. This is the raw score on the y-axis. And here you can see that there's a significant decline in this set of over 50,000 antibodies as they mature. And it held true over 3 different donors when we adjusted for their HLA. So that was a very exciting finding, but we decided to look more closely at the T effector and the Treg epitope content. Here you can see by taking the T-regitopes out, we start seeing a pattern where actually T effector T-cell epitope content goes up, which is not surprising considering that the CDRs are adjusting for higher affinity. And furthermore, if you remove any epitope that's human-like, we also see that the item score goes up, meaning there are more truly non-human T-cell epitopes in those CDR molecules as they mature, as their somatic hypermutation index goes up. And finally, when we look at Treg epitope content or T-regitope content, we see a decline. So as antibodies mature, what we're saying is that T-regitope content decreases. What does this mean to you? It means that when you're isolating from a single B cell, say a new COVID antibody, you want to carefully check for the presence of T-regitopes, because as that antibody's matured, it's removed the Treg epitopes, and your drug may be immunogenic. And I'll let Frances illustrate that for you in her talk. So there's the summary of what I just showed you. And now I'm going to turn the floor over to Frances Terry, who's going to give you an Esprit Demo. Take it away, Frances, and thank you.
Thank you, Annie. In our remaining time, I would like to show you an example of how to use an in silico platform to evaluate epitope content in therapeutic antibody candidate molecules on the Interactive Screening and Protein Re-engineering Interface, Esprit. This is a secure online platform developed and maintained by Epivax for the purpose of immunogenicity risk assessment and mitigation, and it includes tools for the analysis of therapeutic protein and peptide sequences. Today, we'll focus on antibody analysis. On this menu, an antibody immunogenicity report can show you a detailed map of the predicted HLA ligands in your input sequence. Here we will use a subset of 10 of those 50,000 variable heavy and light chains from the natural antibodies from yellow fever vaccine exposed individuals shared by the KU team, the file called YFV demo, starting with a summary HTML report using the enhanced Chothia CDR enumeration scheme from Andrew Martin. A summary table, shown here on the top right, displays high-level information about the input sequences, including their names, lengths, number of predicted epitopes or Epimatrix hits, and Epimatrix scores, both raw, shown in the second to last column, and adjusted for the presence of T-regitopes, shown in the far right column. The greater the difference between these two scores, the more T-regitope content is present in the antibody. The detailed Epimatrix report shown here on the left includes an assessment of each 9-mer frame for binding potential to the globally representative HLA-DR supertype alleles shown across the column headers, where darker blue shading indicates higher likelihood of binding. The last two columns in this report describe the percentage of human antibody isolates that contain a given 9-mer in the ABDB or antibody database frequency column, and the number of human germlines cataloged by the IMGT that contain the 9-mer in the germline count column. As you can see, some 9-mer frames are predicted to bind to more HLA-DR alleles than others, and 9-mers with promiscuous binding profiles are called epibars for this horizontal color pattern, and their sequences are highlighted in yellow to call attention. We believe that if these sequences are found in relatively few human antibody isolates and IMGT germlines, they represent a greater immunogenic risk. In contrast, known T-regitope sequences are highlighted in green. And while these sequences may be predicted to bind to many HLA alleles, we believe and have observed that they are found at high frequencies in human antibodies and germlines, which is likely a driver of their non-immunogenic profiles. Finally, at the bottom here, you can see the beginning of a CDR indicated in red font. Notably, the specific human germlines containing each 9-mer are listed and hyperlinked in each of the interactive dropdowns in the germline count column. To plot a set of antibody candidates based on their predicted ADA response rates, according to our regression model, we can select the high throughput antibody characterization and ADA prediction tool on the antibody analysis menu. This tool allows us to view a visualization of immunogenic risk for multiple candidate variable heavy and light chain combinations, like our yellow fever vaccine mAbs, here paired by name. The resulting antibody immunogenicity prediction heat map shows the variable heavy chains on the vertical axis and the light chains on the horizontal axis in ascending Epimatrix score order. Each colored cell on the grid is produced by the pairing of identically named sequences, as you can see here, and linked to a line of detailed information in the categorized immunogenic potentials table, where you can view the T-regitope adjusted Epimatrix protein score of the candidate, its T-regitope content, and predicted ADA response. We've highlighted the example antibody 635-31-747, which we'll call 747 for short. And now, even though 747 is a natural human origin sequence, it contains high effector T-cell epitope content and low T-regitope content, which would constitute a high risk for antitherapeutic immune response. According to our regression model, this antibody would have the potential to drive ADA in about 38% of exposed individuals. We can also generate an ADA prediction heat map for a set of variable domains where specific pairings are unknown or yet to be selected using the all versus all setting. The resulting heat map here calculates a score and predicted ADA value for each variable heavy chain paired with each variable light chain, regardless of name. And the cells in the grid are still hyperlinked to detailed information in the, albeit much longer, categorized immunogenic potential table. You can see here that in this set of pairings, there are several that fall into categories with low effector content and high T-regitope content, or with low effector and low Treg epitope content, both of which are low risk categories. It's worth mentioning that this level of variation is not unique to our example set of yellow fever vaccine antibodies. And using our antibody immunogenicity scale, I will show you how a smaller set of these sequences compare to natural antibodies generated by a survivor of the 2014 outbreak of Ebola in Zaire using this file called YFV Ebola comp for comparison. The antibody immunogenicity scale is a visual representation of immunogenic risk based on the ADA rates predicted by our regression model. On the left side of the scale, we show observed ADA rates to well-known mAbs that have been in the clinic, and on the right side, the predicted rates for our input sequences. In this case, you can see that the two Ebola targeting antibodies, shown here, have a range of predicted ADA rates, and the higher scoring antibody is similar in immunogenic potential to one of the yellow fever targeting antibodies in our previous example. Now we know that immunogenicity is a result not only of the overall global epitope content, that's the basis for the scores that I've shown up to this point, but can also be driven by localized hotspots within a candidate therapeutic sequence. The cluster analysis menu on the Esprit website can identify these regions, and once we find them, we want to understand whether any similar sequences have been previously reported in the literature as MHC ligands for T-cell epitopes. And we can perform this analysis on Esprit using the homology analysis menu. Here we'll submit epitope clusters to a homology search against the Immune Epitope Database, or IEDB. Users have the option of filtering the matches based on MHC class, here restrict results to class 2, origin of the sequence, restrict results to human epitopes, or HLA restriction, here restricting assays to human host alleles as opposed to model animal MHC, as well as the similarity of the matching sequence. And here we'll use our default setting of class 2 response and 80% amino acid similarity, which is slightly looser than strict identity-based searches and allows for conservative mismatches in amino acids, since we know that these often exhibit similar binding patterns. The summary report shown here is a condensed review of previously published related epitopes, where the number of times a related peptide was reported as positive or negative is tallied in an interactive summary table. We've selected one of the yellow fever antibodies, which we will call 22 as an example. And clicking on the alignment link in the far right column will show the results in greater detail. As you can see, if we view the alignment report for the first cluster in the variable light domain, there are multiple related sequences that have been published in the literature and curated by the IEDB. Now, the far right column shows us many positive elution records, indicating that these peptides are naturally processed by antigen-presenting cells and presented by MHC. Moving left, the presence of both positive and negative MHC binding records might indicate that these epitopes are HLA restricted, while the presence of both positive and negative T-cell assay records can indicate HLA restriction, disease dependence, or differential response based on the outcome measure, which might be cytokine release or proliferation. And the details from each publication can be reviewed by clicking the link in the epitope ID column, which goes to the IEDB record for the published peptide and carries no confidential information about the input sequence analyzed on Esprit. Now, in cases where information on related published sequences is either absent, incomplete, or maybe inconsistent, it's often helpful to categorize the T-cell epitope clusters based on humanness using the Janus matrix algorithm. As you'll recall, this algorithm searches the human proteome for sequences with identical T-cell receptor-facing residues, shown here in green, and compatible HLA-facing residues, shown here in blue, on the assumption that the T-cell sees the whole complex of MHC and peptide, and these might be cross-reactive sequences. The class 2 Janus matrix results for our yellow fever variable light chain shows us the number of human matches, which is the number of human proteins that can be related to the input epitopes through shared TCR-facing amino acids, as well as the Janus homology score, which is the average depth of coverage in the human proteome per Epimatrix hit in an input peptide. For each of the 4 clusters in the variable light domain, Janus matrix human homology scores greater than 3 are considered elevated for input sequences of human origin. And for this chain, we can see that one of the clusters contains a T-regitope. You can see the difference between the raw and T-regitope adjusted score, while another is highly human, shown with this elevated Janus human homology score. And the remaining two clusters overlap antibody CDRs and are comparatively foreign. If we take this high-scoring variable light chain cluster as an example, we can see that the Janus matrix human homology score is elevated. Looking at the detailed matches, we can see multiple human sequences, including a known T-regitope, shown here in green, as matches. This kind of profile suggests at least some bias towards immune tolerance and may partially explain the mixed responses observed in the previously published epitopes we looked at a few slides back. In contrast, if we look at the next cluster in the variable light chain, we can see that the Janus matrix human homology score is below the threshold of 3 that would normally indicate some potential for homology-induced tolerance. Now, we can see multiple HLA promiscuous epibars in this sequence, highlighted in yellow, only one of which has many human matches. The majority of the human matches actually occur in this frame with relatively few Epimatrix hits, indicating that any tolerance for this sequence might be HLA restricted. I hope that this snapshot has illustrated how, considering both the humanness of the T-cell epitope clusters and the phenotype of T-cell responses to any related previously published peptides, in context with the indication and mechanism of action of the candidate therapeutic, can help us to better understand its immunogenic risk. And now I'll hand it back to Annie to summarize.
Thank you, Francis. In summary, we can improve on nature. Basically, what we've shown you today is that carefully trained in silico tools can be employed to predict immunogenicity and tolerance. Predicted effector and regulatory T-cell epitopes can be validated in vitro assays, the IVIP and tetanus toxin bystander assay, respectively. Some pathogens contain human-like epitopes. It's crucial to avoid including these self-like epitopes in new vaccine designs. Furthermore, the presence of these self-like epitopes likely reduces their immunogenicity and efficacy. Removing these epitopes from infectious or cancer vaccines may improve the immune response. In contrast, self-like epitopes, or Treg epitopes, may be removed from what are considered mature antibodies. All is not lost. In silico tools can address and improve on nature. These advances, as described in today's webinar, can be applied to developing better biologics and vaccines.
Thank you for sharing your time with us today. And thank you to all of the team members at Epivax who made this presentation possible.